First of all, I'm very excited to be here to discuss this topic, because I believe this is one of the most important topics that we are facing in health, mental health, but humanity in general. It is one of the most important questions we need to address if we want to promote mental health in the best way and also treat mental diseases. If we address these important questions, we're going to have novel tools to have a better life for everybody and to prevent or cure many devastating diseases for which there is no cure at this moment. So we have started with an Italian title, and in the Italian title, we focused on the idea of talking about how trauma disrupts a positive, good development and integrated development of the brain and of the individual. But I'm going to try to give you a higher level view, a more complex, hopefully, view of this problem, the question, which is how development shapes our brains for life. And that, as I'm going to show you and as I'm going to argue based on our data, that is about learning, memories, and how the biology of the brain shapes these functions. And of course, this includes also how early trauma shapes the traumatized brain. But it's not only about that. All right. So we have heard already in the morning from the talks that we had in the morning, in particular from Diana, that there are a number of important symptoms that childhood trauma leads to, and they are protracted for the entire life. And these are, of course, the medical symptoms that she greatly illustrated. There are psychological symptoms, including post-traumatic stress disorder, anxiety disorders, depression, negative self-perception, addiction, attachment disorders, dissociations, avoidant behaviors, hypervigilance, and emotional distress. More than that, just to give some of the important ones. And these psychological symptoms include cognitive symptoms, like impaired brain developmental milestones, learning and academic challenges, and we have seen that also this morning. There are also physical symptoms. I'm not going to repeat that, but certainly they lead to a number of medical problems, these early traumas of children. We also have heard that there are studies showing us what biological changes underlie the childhood traumas and how these biological changes in the brain created by the traumatic experience change the brain itself, the biology of it, and this is very persistent. And I'm not going to talk about a lot of these mechanisms because there is a great extended literature demonstrating these mechanisms linked to trauma. I'm just going to mention a couple of them. One is the disruption of the HPA axis, hypothalamus pituitary adrenal gland axis, and we're going to talk briefly about that. And the other is certainly DNA changes and epigenetic changes in general, which are induced by the experiences. And these are changes in gene expression, not so much in the type of genes which we inherit, but the regulation of how genes are expressed. And these changes in gene expression can be long-lasting up to a lifetime. I'm not going to get into other details here, but as I said, there is a large literature, both in human studies at the level of behaviors, at the level of biology, at the level of medical tests that has informed us of what traumas, childhood traumas lead to. So what I'm going to argue, based on our data, and I'm going to get into those in a minute, is that it's more than early traumatic experiences. It's not just about traumas. Traumas, of course, have an impact that is great for all those diseases that are linked to the traumas and the upbringing. But the way we need to look at it is not only to understand what traumatic experience lead to, is to understand what learning in early childhood does to the brain and to the individual. It's a higher level view because it's not about one type of experience. It's also the experiences that the traumatized brain didn't have. And I think this issue came up with a question, so let's dive into it. And I'm just going to show you in the next slides that our study showed that brain development does not occur by default. I heard in one talk about it's not just about biology. I think this biology meant default processes of development. Of course, it's not that. But everything that experience does to the brain translates into the biology of the brain. So brain development does not occur by default, not according to programs. There are certain programs, but these programs are regulated by experience. Experience shapes how the brain develops. It's not vice versa. It's not the brain develops and therefore we can do certain things or not. No, experiences control brain and individual development. I'm going to show you, based on our neurobiology research, that experience in early development creates mental schemas that are different than the schemas that are used in adulthood. On these schemas, we, as individuals, sculpt and build our behaviors and psyche throughout life. So it is about learning and memory. So let's dive into learning and memory. I'm going to give you some definition. I'm going to tell you what's known, a little bit what's known about the molecular and biological mechanism of learning and memory in adulthood, and then we'll go to development, early development. So learning is the acquisition of information, and memory is the storage of information. The retention of learned information that allow us to recall previous experiences, and based on that, change our behaviors. Memories are essential for life. Without memories, we will not be able to survive because we will not be able to recognize danger. We will not be able to recognize the best resources. And therefore, learning and memory is an essential function for life. Memories make also us who we are. They are the most important component, I would argue, of our identity. Just think about cognitive impairments, memory pathologies, in which memories are lost, cognitive functions are lost, we don't know who we are, where we are, what we have done, and everything collapses. So memories are also the major component of our identity. There are different types of memories. I'm going to go fast here because I think also, especially the students, have seen these slides from me in the past. One classification is about the duration. We have short-term memories and long-term memories. Short-term memories are the memories that last for minutes, and long-term memories, they last for days, months, even years. Very long-term memories last for a lifetime. How do we get from short to long-term memories? First of all, there are different types of memory that can be short only, like working memory, for example. Long-term memories become long-term with two types of mechanisms. One is emotional regulation, emotional charge. Emotions can be positive or negative, but they have to be there in order to store long-term memories. And the second is repetitions. Another way of classifying memories, which is not opposite to this, is also, in addition to, in the long-term memory areas, we have several types of memories that we can group in two major groups. One is called explicit memories, and in these memories, we have the so-called declarative or autobiographical memories in humans. These are the memories that we can talk about. So if I say, what did you do last night? You are gonna use your autobiographical memory. What did you do last Christmas, or whatever it is? Who did you meet last week? You're gonna use these autobiographical declarative memories. These memories are recalled consciously. So you are conscious of them. And they have features: conscious recollection, they are verbal, they have deliberate recall, they are more controlled, and they have context, time, and space in it. These are the memories of facts, people, and things, and places. They include episodic and semantic memories, the memories of that one time, last night, which is unique, and semantic memories, which is general knowledge of something. If I say dog, you know what I mean. How did you acquire that general knowledge? And what is that type of memory? It belongs to these explicit types. These memories are processed by the medial temporal lobe. It's a circuitry in the brain that has been studied very much in terms of these type of memories. And particularly, as we will see, one of the regions that has been studied a great deal is the hippocampus. On the other side, we have the implicit type of memories, or non-declarative. These are memories of how to do things. So for example, you learn how to ride a bike, or to drive a car, or to tie your shoes, or to play piano, or to ski. Those are memories that you don't need the verbal actions to recall and to explain. You do that implicitly. These are, for example, procedural and skills and habits memories. There is also priming, subliminal memories, non-conscious memories. And there are a number of very simple type of learning and memories like reflexes. For example, non-associative learning, habituation and sensitization, and so on. And all these memories, which are all implicit, so non-conscious recall of it, belong to this umbrella of implicit, non-declarative types of memory. These two systems are independent. They can work independently. There are cases in which the explicit memory system is impacted, like in Alzheimer's disease. The implicit memory system works. And a number of cases have shown that this system can work independently, but if they are both healthy, they actually cooperate in a healthy brain. So medial temporal lobe and regions of emotional regulation are always working together to form long-lasting episodic memories, for example. So we're going to talk about the episodic, explicit type of memories. This is the memories we are working on for 30 years and more. And these memories, as I mentioned, are autobiographical, episodic, and they contribute to our sense of self and identity. In humans, is that event of that evening, of that moment, with all the people there, all the stimuli, whatever you heard, whatever you smelled, whatever you met, whoever you met, and whatever you did. And these memories, as I mentioned, are mediated by the medial temporal lobe, in which the hippocampus is particularly studied and plays a critical role. If there is a lesion or an impairment of hippocampal functions, these memories do not form and store. These type of memories are very well reproduced in rodents. Rodents have memories of who, what, where, and when, conspecific, objects, spaces, and time. And they use the same memory system in which the hippocampus play a critical role. So we have an evolutionary conserved system here that we can study in rodents and get to the biology of it. What do we know about the molecular mechanisms, the biological, molecular, and cellular mechanism that happens in this medial temporal lobe during the formation and storage of long-term memories in adults? We know quite a bit. In the last 30, 40 years, there has been a tremendous progress in the identification of the molecular mechanisms required to form and store long-term memories. The hippocampus has a temporally limited role, but it is essential. After learning, the hippocampus is activated together with cortical regions and regions that process emotions, and all together, this activation that is coordinated by rhythms lead to biological changes in the circuitry. And these biological changes change actually over time. So this is exactly what we study. We study these biological changes after learning that over time lead to memory storage. There is a process that happens after learning with these memories. So episodic memories are not stored immediately. Like you are forming now a new episodic memory, this memory is fragile for quite some time. And if we interfere with either pharmacological or new learning or some physical interference during this time of fragility, we will block the stabilization process of this long-term memory. And the memories are going to be more or less lost. The earlier the intervention, the more is the deficit. This process of stabilization of this fragile memory trace is called consolidation. We are interested in understanding the biology of consolidation. We know from many studies in all species that we have tested that consolidation requires a cascade of molecular changes starting from mRNA translation to gene expression to a number of modifications of proteins that lead to biological changes that are persistent in the brain, and that is parallels the storage of memories. And of course, as I mentioned, these studies have been particularly focusing on the hippocampus and also in some cortical region like prefrontal cortex, in which this reverberation of activity and molecular changes lead over time to memory consolidation. And there are two processes ongoing, is actually one, but there are two definitions of memory consolidation processes. One is these biological changes, gene expression, let's say, in all these, the aspects. And the other is system consolidation, which takes a little longer, meaning if I interrupt gene expression, I see an effect right away, but if I wait in the rodents a couple of weeks, I'm not going to see, or a couple of days, I'm not going to see any effect. But the system consolidation, which is these biological changes occurring throughout a number of brain regions, take more time. They take weeks in the rodent. They take years in humans. So we're interested in understanding all these changes. And as you can imagine, it's a very complex question. Also because biological changes are not the same in all brain regions. They differ in the different regions. They come on and come off at different times. And they last for, as I said, a number of weeks in rodents or months, years in humans. There is another phase of fragility in memory, and that is after we recall the memories. In some conditions, especially if the memories are recent, when we recall them, even if they have been consolidated at the biological level, they become fragile again for a limited time, limited temporal window. So they return to a fragility state. If we apply interferences, we're going to stop this restabilization that is called reconsolidation after the recall of the memory, and the memory is lost. So we have actually here a tool based on the physiology of the memory that we can use to weaken very strong memories, such as memories of PTSD, traumas, or addiction, by recalling them and trying to decrease their strength. It's not so simple, but we have evidence that it's possible. So consolidation is after learning, reconsolidation is after memory recall. Not all recalls lead to reconsolidation. We need to know the conditions to use. Then another level of regulation of the long-term memory formation and storage is modulation. As I mentioned, episodic memories become long-term, and they can last for a lifetime. We all have examples of one episode in our life that we can recall from many years ago, and they can last for our entire life. This is because modulation has become, has been part of this memory, modulation by emotional charge. As I mentioned, emotions can be very negative or very positive, but they are needed in order to store long-term these representations. I'm going to talk as an example about modulation of stress by stress, and that is based again on a number of studies showing that emotionally arousing events are better remembered, and we all can relate to that, suggesting that the hormonal system activated during these experiences, hormonal stress hormones, can modulate memory consolidation, and that is what happened. It has been shown also in animal models and in parallel in human studies that adrenaline and cortisol are required for memory consolidation of single events. They are required, and they also enhance long-term memory. Regions like the hippocampus and the amygdala, the regions that regulate emotional valence, are known to be very important for memory consolidation. If you impair the activity of these regions, there's going to be no memory consolidation. And these regions express high levels of receptors for stress, noradrenaline as well as glucocorticoids. So modulation is key and works together with the biological changes occurring in the medial temporal lobe, therefore amygdala in this case, as an example, through neuromodulation is working to change the biology of all those regions required for memory consolidation. So memory consolidation of emotionally valent experience is going to be different. It's going to be a different biology. It's going to give you different changes in those regions that store long-term memories. And the same is for glucocorticoids. In particular, glucocorticoids have been studied at the level of traumas because they have been found to change with traumatic learning and memory. So this will go back to the HPA axis that is regulated by learning, leads to the release of noradrenaline and glucocorticoid hormones. If the level of stress is very high, the level of glucocorticoids release is going to be higher, and this is going to engage different type of receptors, which are the glucocorticoid receptors, rather than mineral corticoid that are engaged when the levels are lower because they are low affinity receptors. So just to summarize the effect of stress, stress is good, just doesn't have to be too much. Stress is necessary. This is a curve that has been shown to characterize performance in general, including learning and memory. If you don't have any level of arousal or stress, there is no function, there is no performance, there is no feeling of well-being. You need some level of these hormones to feel alive and good and perform. Clearly, then these levels reach a plateau. Over that, the performance decreases again, meaning high stress is going to impair performance and the ability to do things. So on the left side here, we have the adaptive learning and memory system regulated by emotions. On the right side, we have the maladaptive, so-called maladaptive. In reality, everything is adaptive. Let's remember that everything that the brain does is to protect. But of course, the expense in some very extreme case is going to be a maladaptive response of the individual, okay? Depression is adaptive, then is a problem, of course. Okay, so we have studied the biology in the adaptive, the left side of the inverted U curve, and the right side of the curve, and the maladaptive memories. The inverted U curve has been reproduced in rodents. We work with mice and rats by changing the level of stress linked to the event of memory. We have found exactly the inverted U curve in the behavior, and by studying the left or the right side, we have looked at the biology in the medial temporal lobe of those animals, as well as the behaviors, and we have found that the biology of the hippocampus and prefrontal cortex involved in forming and storing adaptive versus maladaptive or traumatic memories are different. The biology is different. The circuitry involved is different. Traumatic memories also have different behaviors, are resilient to extinction, for example, and are very hard to undergo reconsolidation, the disruption after the return of the memory. So here for the therapist, I actually forgot I wanted to mention, I also train as a psychoanalyst, so I'm happy to engage in those discussions. For the psychoanalyst, we need to know all of this in order to know how to intervene, because the talking cure is about recalling memories. Are we recalling memories, and can we work on these fragile windows? How about traumatic memories versus other memories? There is quite a bit that is known now. I cannot get into the details, but I think those are very useful information. And of course, as I mentioned, the behaviors of those memories, the traumatic memories, is very different. So let's talk about now learning memories and memories in children in early development. What I told you until now, and I didn't go into all the molecular mechanism, but just to say that there is a lot known in those regions, in those circuitry over time. Of course, those mechanisms, biological mechanisms, have been discovered in rodent systems, because we cannot use so much human brains unless they are post-mortem, of course. But the information from the biological system of the rodents can inform human studies and vice versa. So I think that that is a very important cooperation that has to happen more. Okay, so let's go to children. Is learning and memory in early developmental phases working with the same mechanisms? Does it use the same systems? Or are these different types of system biology and memories that happen? And of course, here as well, we have more adaptive or happier or positive memories, and we have also traumatic upbringing. Like in adults, we expect this to use different biology, but we don't know. In fact, a number of studies have identified the biological mechanism of traumatic early life memories, but they have not studied normal learning and memory. So a number of years ago, about 10, 12, this became a very important question for me. In my lab, we have studied the biology of memory in rodents using adults. And now we face this question: what happens to the biology of memory in babies, in baby rats, in baby mice? What's the biology? What's the circuitry? What's the behavior? This was, of course, informed also by my psychoanalytic training. So if we look at some biological parameters, one is the development of the brain, as you can see here, from fetal to postnatal to, a few years after the individual is born, and these are humans. You see already from the shape how different they are. So if I take the postnatal, I think it's this one, can read the numbers, but, and here is very small, but if you read about the discovery of the morphology and the biology, it's very different compared to the brain of a few years later, 2 years of age, or young adults. Very different. The brain of an infant is not a small adult brain. We know that. But I keep repeating this to myself, because we cannot extrapolate what we know from adult behavior and biology and medicine to the babies. It's a different organ. So we need to understand the biology. And on this side of the slide, you can see just one parameter, which is synapse formation, demonstrated by Golgi staining at different ages. This is the newborn. This is 3 months. This is 6 months. This is 2 years. And then after that, there is synaptic pruning, a reduction of the number of synapses. So as you can see, again, over time, postnatally, but of course prenatally as well, there are huge changes happening in the brain. And this is the result of the interaction of the genetic background. We cannot forget about that. The genetic background is going to give us the possibilities, but mostly is the result of experience. If you block experience, all this does not happen. So what happens in infancy? Well, there is a very interesting phenomenon, that is early life memories are formed, but they are very rapidly forgotten, very quickly forgotten. I'm talking about episodic in humans and episodic like in rodents. So this forgetting is believed to explain infantile amnesia, the inability as adults to recall the first 2-3 years of life. Anybody remembers what you have done in the first 2-3 years of life? Sometimes there is an exception, and it's always interesting to go back to that. By and large, we don't. And because during these first few years of life there is a very rapid forgetting, this was believed to explain the fact that then we do not have memories of those early years of life. So on the left side here, you have a graph showing infantile amnesia in humans. So here they ask subject, human subject, in adulthood. Oh gosh, very sensitive, sorry. They ask here, human subject, how many memories they had, autobiographical memories. And as you can see, in the first 2 years of life, there is basically 0. Then maybe somebody said, I remember at 3 years old. But then they grow, they increase very, very steeply after the year 4, 5, 6, 7, 8, and then they plateau. These human memories are rapidly forgotten. And this rapid forgetting has been reproduced in rodent animals. So if I take rats, like in this experiment Campbell and Campbell did in the 60s, they took rats at postnatal day 18 or 23. Postnatal day 18 in rats correspond roughly to 1.5, 2 years old in humans. 23 is juvenile, 4, 5 years, 6 years in humans. And they compare the learning at this very infant age to the learning at later ages, like pre-adolescence, P38, P54, after adolescence or in between, in the middle of the teenager time, and P100, which is young adults. So they took different groups of rats at different ages. They gave a learning event, one episodic learning event. This is called inhibitory avoidance. And in this task, the animals learn that there is a context in which they're going to get a food shock, so they learn to avoid it. Very simple. We do that also as humans. Not with the shock, but something else, maybe. Okay, so they trained them all in a similar way at different ages, and they tested immediately after learning. And as you can see here, all these groups had comparable memory performance. And then they started testing at different times. And here is the memory of the postnatal day 18 infant rats, the curve of it, 23, and then 38, 54, and 100. The infant rats forget very quickly. A little bit later, they have more memory retention, and then after that, they have a very long-lasting memory retention, which is comparable. So we have a problem here to address, because a lot of literature, psychology, psychoanalysis, even common sense, right, tells us that experiences, this is a quote from Freud, so not yesterday, experiences of the earliest days of life, years of life, leave persistent traces in our mind, which change our behavior throughout life. And then he commented about infantile amnesia because he saw in the patient that there is no recollection of memories of the first few years of life. So there is a paradox, right? If we lose those memories, how can we explain that they influence our life forever? Something is not addressed. Several hypotheses have been proposed to explain this infantile amnesia or rapid forgetting. Freud was the first one to give an explanation, and he said that this is repression of something that has caused too much excitement, negative excitement, probably. Disturbing content of sexual nature. Cognitive scientists later say, oh no, it's the lack of language skills and lack of sense of self. If I don't have that, I cannot have a conscious recollection. However, rapid forgetting happens in rodents. So we need a neurobiological explanation for it. Okay, neurobiology says the hippocampus is too immature. The hippocampus-dependent memory system, medial temporal lobe-dependent memory system, is immature, is unable to consolidate the memories, and therefore they are not stored long-term. The hippocampus is offline, unable to work yet. Developmental hypothesis. Another said, maybe not. Maybe those memories are stored somewhere, so we can somewhat give an explanation for the fact that they have a long-lasting effect. However, they cannot be retrieved because the retrieval system is not mature yet and cannot recall those memories of infantile experiences. And this undergoes under the retrieval hypothesis. So we have studied all these questions in our rodents, and the approach is to use rats or mice, depending on the tools that we need, and hopefully also to see cross-species confirmation. We use episodic type of learning, episodic-like type of learning, and those are aversive, like the mouse or rat is placed into a box. I don't know what happened now. Oh, only to me. My screen disappeared. Aversive type of learning, the mouse or rats avoid a context paired with a food shock, inhibitory avoidance. We also have non-aversive type of learning, which is novel object location. These are, again, memories that depend on the medial temporal lobe because are spatial memories, and they recognize how, where, and how we move the object in space. We have other types of learning that we use, social learning as well. And we interrogate the brain of these baby rats or mice. We want to understand what type of biology these regions start with and what type of changes they undergo with these learning events in infancy. So we have addressed a bunch of, I think, relevant questions. The first one is, so are these memories gone? The infantile memories that have their happy forgetting. Are they gone from the system? Do we lose them? Or are they somewhat stored in some format, and that's why they can influence behavior throughout life? As I mentioned, most of the studies were supporting the developmental hypothesis. That is, memories, long-term memories are not stored in those early age of life, and therefore we lose them. So we did simple experiments. We use, again, inhibitory avoidance to test the memory at different ages. So we reproduce what I already showed you. In this case, it's postnatal day 17 instead of 18, postnatal day 24 instead of 23, and 80 to 100 young adults. When we compare that, you can see the performance curve in blue. Those are infant rats. They have a memory immediately after learning that is comparable to that of the other ages, but 30 minutes later, 90% of the memory is gone. We replace the animal back into the box. They do not remember that they had a food shock. They just go right in. The other two curves, orange, which is postnatal day 24, juveniles, and white, which is adults, they have a long-lasting memory. And they're even more long-lasting, of course. We stopped here at 7 days. So we tested a number of protocols to see whether we could get back the memory that is forgotten here in the blue groups, in the infant groups, by re-exposing them to the context. 1 time, 3 times, number of times, many, many times, the memory did not come back. We gave them the food shock in a different context. The memory did not come back. But if we combine the context memory recall, and a few days later, we gave the food shock in a different place, now the memory came back. See, the blue line is comparable to the adults. Very strong, sustained, we kept testing and it was there, and context specific. This is not generalization. When we tested these animals in another context, they had no memory. Only the context that they have seen in infancy was the context they recognized. So, the episodic infantile memory is not forgotten, is not expressed, but is there, and we can get it back with certain types of reminders later in life. This can happen even in adulthood. We waited until adulthood, we give certain reminders, memory comes back, and there is more to it. A number of studies, us and other labs, have then used what is called artificial reactivation. So you can label the cells that are active during infantile learning. There are mice that offer the technique, this technology. You label them, and you make them express an exogenous receptor that you can activate or deactivate whenever you want. So if you do that and you label the trace of the infantile memory, you wait some time, and then you reactivate it, you get the memory back. So we know that there is cellular components that you can reactivate artificially to get the memory back. So this confirms the behavioral data. There is a physical substrate that you can use to do that. So memories are stored. They are not expressed. They can come back. Question 2, is the hippocampus-dependent memory system involved? Or are they right by saying that the hippocampus is not online, is too immature, doesn't work at all, and therefore there is some other brain regions involved in this development, in this memory, sorry. So what we did, it was to inactivate the hippocampus and then give the infant learning and then give the protocol that takes the memory back, and there was no memory. So this, I'm not going to show you all this data, it's a bunch of experiments. These results clearly demonstrated the hippocampus is needed to form these infantile memories that are not expressed but stored. Needed. This was changing completely the way this type of learning and memory in infancy was seen until then. 10 years ago, it's not so many. So the hippocampus is required to form and store these memories, and the hippocampus is part of the explicit memory system. Of course, it's not mature as an adult hippocampus, but is required. So we went into the hippocampus and asked, what type of biological changes happens in the infant hippocampus with this infant learning? And what we found is that the biology of the infant hippocampus is different, that we sort of know also, but also the changes involved in storing this memory that are recruited in infancy, in the infant hippocampus, are different. These include the increase in synapse numbers and the maturation of certain type of synapses. These mechanisms do not occur later on in the hippocampus with new learning, only in infancy. More importantly, what we discovered is that there is a recruitment of biological mechanisms in the hippocampus with this learning that are mechanisms typical of critical periods. Critical periods have been studied in sensory systems like vision, in language, but the biology of it, of course, was studied in sensory systems, and particularly in the visual system. The newborn, when we are just born, our vision is not good. It increases through visualizing, through the stimuli, through the visual stimuli. The visual stimuli mature the visual cortices that shapes in a way that then works perfectly, or adapt in the best way, in the adult type of way. During this development, through experience, visual experience, there is a temporal window that is very defined, and that uses a different biology, certain type of mechanisms, that promotes the maturation and the development of the system. Once the system is developed, this biology is turned off. This temporal window, which is called critical period, is very important for the development of the function. So let's say a baby is born with a cataract or 2 cataracts. The visual system with 2 cataracts, if we close the eyes, not gonna develop. But if we reopen the eyes within the critical period temporal window, we can recover the function. We can recover a perfect vision. So if we remove the cataract in that period. But if we wait after this temporal window is over, the vision is going to recoup a little bit, but not in the same way as it developed through the critical period. So critical periods are essential temporal window during which experience shapes and install the function in the brain. So we found that this mechanism of critical periods, I'm not going to list them, but there are a number, biological mechanisms, happens in the hippocampus of baby mice and rats. So critical periods that, as I said, are this temporal window that develop the functions. And the brain develops over time from the most simple to the most complex function through critical periods. So at birth, we start the critical period of sensory systems. Until we don't have sensory systems developed, we cannot do something more complex, like, for example, having an episodic memory. Then we have motor and language development, and then over time, we're going to have higher cognitive functions. So the critical period work in sequence. If we impact the first one, everything else is going to be impacted because the first one leads to the second, which leads to the third. So cognitive function is the result, the end result of a number of critical periods. So we proposed, based on the fact that we have a very limited temporal window for learning and memory in infants, they use mechanism of critical period, and the hippocampus not only is required, but we found that the learning experience matures the hippocampus. It matures biologically, and it matures functionally. So if we give more experience to the baby rat or mouse, more experiences, repeated experiences, they develop the function. You can measure that. So we proposed that the hippocampal medial temporal lobe dependent memory system undergoes a critical period, very much like sensory systems do, very much like language does, which means that the medial temporal lobe develops through experience. That's the hypothesis. Let's test that. So the hypothesis says the system is not developed yet. Like a blurry vision when we are born, the early hippocampus doesn't know how to express long-term memories, but it is required and respond to the experience. And through experience, it matures the function of long-term memory. So do you see? This is the opposite of what we are reading in the textbook. Development happens, and that's why once it's mature, we have the function. No, function happens, and it matures the system. In fact, if this is true, as I said, repetition of experience should lead to maturation of the function, long-term memory. And if this is the case, there is another important question. Does it mean that maturation is experience selective or specific? So we tested that. And very briefly, doesn't matter that I show you all the details. We gave repeated experiences now, episodic experiences, day 17 and day 18. If I give one, day 17 or 18, they have no long-term memory. If I give one at day 17, one at day 18, now repeated, memory performance is there. There is a very strong memory when I measure after that. So we can do more than that. How about testing in a different context? Is this memory context specific, or is it more generalization? So we tested them in a different context, these mice and rats, and guess what? There is no memory for the other context. It's context specific. Now, what if I give the first learning event in context A and the second in context B? What type of memories do I get? Do they talk to each other? The answer is yes. The first memory allows the second memory to give a long-lasting performance. These memories, they can discriminate the context I just showed you. If I give two context A, they have no memory for context B. But if I give A and B, now they have memory for A and B. So it means that the type of memories that we create through experience, through development, through developmental experiences, are specific for the type of experiences we encounter. And in fact, more than that, if I give two types of experiences that are very distinct, not congruent, for example, one inhibitory avoidance with the food shock in the context, and one object location, these are memories processed by the same system, but they're very different, non-congruent. Now I have no memories for neither. There is no memory for either. So congruent memories talk to each other. Non-congruent memory, they do not talk to each other in the hippocampus-dependent memory system. So just to summarize this, infantile memories are stored in a latent form, perhaps implicit, perhaps unconscious. We cannot ask the rodents, but that's what I think. The ability to explicitly express long-term memory matures through repetition, through experience of congruent learning. Maturation of cognitive abilities is experience-driven and is specific for the type of experience. So one last question, very briefly, I'm going to tell you because these are unpublished studies. We're trying to understand why is this happening? Why we have no memories expressed, yet there is a memory trace that is stored until adulthood. So now we give this learning event in infancy, and we are looking in adulthood what they do. And I can tell you in summary that we can get the memory back in adulthood, and also these infant memories influence the acquisition of new memories in adulthood if they are congruent, not if they are not congruent. We can even label the memory traces and see the overlapping when they are recalled and when the little boost of the infant memory is working with the new learning to create this new congruent learning based on that infantile experience. If we don't give the infantile experience, it doesn't happen. In other words, I cannot show you this data, but through labeling the cells involved in the activation with this early learning, the reactivation in adulthood, either by recollecting the memory or exposing them to what is called savings, just a subliminal stimulus, and then new learning, and see that the new learning happened only if they had the infantile memory. We proposed that in infant learning, there is the formation of schemas that support subsequent learning at later ages, including in adulthood. These type of schemas we tested, they do not occur when the animals are adults, only during infancy, early life. So in conclusion, am I too completely out of time? Developmental maturation of brain functions does not occur by default. Experiences encountered during early development, critical periods, mature the functional competence of the hippocampus-dependent memory system in an experience-specific, selective manner. This function is selective for the infantile age, doesn't occur in adulthood. The brain regions involved are, I didn't show you the data, but are hippocampus, anterior cingulate cortex, and prefrontal cortex. I'm sure others, this is what we tested. So we suggest that infant episodic learning builds stable fundamental schemas that are used later on to build congruent learning, to promote efficient congruent learning. And these schemas are based on experiential traces. And in the adult brain, they re-engage part of the same population that were active in infancy, in the infant learning. So I'm going to go through 3 implications, and then I'm going to stop. Latent infantile memories which develop the brain, the system, in an experience domain-specific manner, offer an explanation for individual differences. My infantile experiences have been different than yours. And this is where in psychoanalysis we say the devil is in the details. This is not just about trauma. We're going to get to that in a second. It's about everything we process during the first few years of life, and later, of course, critical periods, they keep building until adulthood. Yeah, adolescence is another important phase. But until we are not fully developed, which is about between 25 and 30 years of age, prefrontal cortex, we're still building on critical periods. Yeah. So developmental learning shapes who we become and how we act, including automatic, implicit, and unconscious responses. Traumatic experiences is one aspect that, of course, impact the brain of those individuals tremendously, because that brain has learned traumas, but that brain didn't learn other things, didn't have other experiences that are key. It's not only what we learn, it's also what we have not learned. That is very important for understanding traumas as well as anything in the development. Developmental experience-based brain maturation underlies the importance of learning, education, policies, life context in mental health. If we want to have a better life, more adapted, and with more abilities and tools for us to go through life, we need to build a better brain. We need to build mental health. This knowledge will inspire question about mental diseases, because neurodevelopmental disorders very likely are the results of impacted critical periods, like autism. We can talk about that. So we can target those mechanisms and specific critical periods to have better cures or better life, even better, and prevent a number of mental disorders. Another point that I want to stress again is that both biological and behavioral changes come from biology, let's not forget that, created by infantile experiences are distinct from those created in adulthood. The baby brain is not a small adult brain. The brain in early development is not a small adult brain. This has enormous implications for neurodevelopmental disorders research and therapeutic approaches. Focusing on development, starting from the earliest phases of life, is the best approach, in my view, to make significant changes, improvements in mental health, and reverse problems in mental diseases. I want to finish by thanking who did the work here. I have only a small group who work on development, but they did, I think, fantastic work in trying to address important questions. I want to mention former and current lab members, of which I did mention very little, my collaborators, and of course the funding that supported our work, and I thank you for listening.